Welcome back, participants. Now we are entering the first technical session of today. Now we start with the oral presentation under the presence of Chairperson Professor Dr. V.S. Vadai, HOD of Microbiology Department at Sardar Patel Mahavidyalaya, Chandrapur, Maharashtra. However, due to the paucity of time, we would request the speakers to limit their respective presentations within 10 minutes. Now, I request Ms. Ashwini Vijaykumar to present her topic on gut microbiome, a companion or foe to health.
Hello everyone, I am Ashwini Vijaykumar. I am happy to share my review on topic of gut microbiome is really a good companion or foe to our health. Introduction. Human body is colonized by numerous microorganisms collectively referred as microbiome with broad diversity distribution from the surface of the skin to the internal organs in a specific range. The word microbiota expresses the association between human and microorganisms originated from Greek. Micro means small, biota means life. The microbial community can be classified based on their interactions, beneficial, pathogenic or neutral. Diversity of microbiome. Microorganisms that occupy the habitat of host are predominantly bacterial groups. Other than bacteria, archaea, viruses, fungi and protozoan were also present and collectively called as microbiota. When this association occurring at gene level known as microbiome, human body will always try to bring back the homeostatic condition even by reviving back original microbiota. Otherwise, it will try to bring the balance by replacing microorganisms which carry similar physiochemical properties like alpha and beta diversity. Major factors that are affecting microbiome are listed below. Age. Colonization of gut takes place before the birth of infant and later gets developed based on the birth process, normal delivery or C-section, then modified through diet, breastfeed or formula milk. Around the age of 2 to 3 years, the stabilization of the gut composition depicts like adult gut with 40 to 60%. Diet. Intake of high amount of western diet will cause immune mediated inflammatory diseases, for example cirrhosis, rheumatoid arthritis, etc. Xenobiotic metabolism. When any foreign compounds like drugs, for example antibiotics are also known as xenobiotic compounds, will cause adverse effects on host health, developing condition called biotransformation. Antibiotic activity. After effects of the antibiotic treatment will leave a short or long-term implications in health. This mainly happens when host getting resistance or breaking the medication cycle. Environmental effects. Smoking alters the level of bacteroids and Prevotella species. Silver nanoparticles in food industry will produce environmental stress which highly affects the diversity of gut microbiome. Next are some of the examples regarding the diseases and disorders caused by dysbiosis in gut are listed below. First one is irritable bowel syndrome. IBS is one of the gastrointestinal disorders where recurrences are common in chronic persistence. Based on the food habits, way of defecation and its consistency, IBS will affect the individual both mentally and physically. Obesity. Many chronic metabolic disorders conditions have been increased worldwide. The main reason behind is that individual being overweight. Diabetes. Type 2 diabetes mellitus is one of the metabolic disorders caused by imbalance of blood glucose level, altered lipid profile by the microbiome. One of the reasons behind today's prevalence of chronic liver disease is non-alcoholic fatty liver diseases. The extended access from gut liver increased other clinical conditions such as obesity and type 2 diabetes mellitus. Alternation of microbiome column shows the variation of the microbiome in different clinical conditions. Next one is lung diseases. Increase in number of respiratory diseases like cystic fibrosis, asthma, chronic obstructive pulmonary diseases, COPD and their symptoms are related to gastrointestinal disturbances like irritable bowel syndrome, reflux between esophageal and stomach. And in case of COVID-19, the virus gets recognized by ACE2 receptor in gut. Gut microbiome is the host defense mechanism against the pathogens that gains access to other organs with their bidirectional pathway. This is the main reason of increase in susceptibility and difficulty in treating people contains health issues with heart, diabetic and other respiratory illness treatment. Chances of reversal of this imbalanced biodiversity of microorganisms can be regulated to some extent, though many research experiments were conducted to study about the disease implications as well as trying to solve the current diseases prevalent all over the world. The interest of providing treatment for diseases through microbiome is currently in study models such as rat, mice, human with or without administration of pre and probiotics. Thus, microorganisms performing their duty to reverse the clinical conditions. And the second one is the fecal microbiota transplant FMT is one of the diagnostic and the treatment method used to control the clinical conditions of the human being. It is otherwise known as stool transplantation, which is possible between the healthy donors to the needy recipient. Conclusion. I would like to conclude my work by stating that gut microbiome is truly a great companion as well as foe to health when it loses its purpose of the existence because of not paying attention to what kind of food we are consuming, because if the food is high in sugar, salt, spicy, additives, preservatives, low fiber diet, high fat content will definitely create pressure on the metabolism of host and disturbs the resident microbial community. The treatment of antibiotic works in bidirectional way because it will suppress or spikes more immune responses. Challenges of this treatment can be concluded such as there is no accurate representation or significant biomarkers that are available for every clinical conditions. Finally, I would like to thank everyone involved in organizing this event and for this great opportunity to share this work. These are my references. Thank you.
Next, I call upon Dr. Bharti Wadekar, who's presenting a topic on application of Bactosine B25 by marine Lactobacillus pentosis B25 as a food preservative in milk.
Good afternoon, everyone. Today I am going to present application of Bactosine B25 by marine Lactobacillus pentosis B25 as a food biopreservative in a milk. I, Dr. Bharti Wadekar, Assistant Professor, Microbiology, IID, ZSCTS, Thakur Shamnarayan Degree College, Kandivali East, Mumbai. Bactosine is a very small peptide which is produced by the extracellular ribosomes, and it is working as an alternative to the antibiotic. This bacteriocin have the three types of classes, and it can kill or inhibit the bacterial strain which is closely related or non-related microorganisms. Then Lactobacillus pentosis is a lab lactic acid bacteria commonly used as a starter culture for the fermentation, and it is the gram positive bacteria, non-sporing bacteria. The first applications as a preservative, the first form or produce niacin and some of the newly isolated novel bactosine were effective as a biopreservative in a certain food system in which the concentrated bactosine preparation or the bactosine producing strains has been used. The lactic acid bacteria strain have their own antagonistic activity to either decrease in the pH or to bactosine production, making them ideal for cheese making and preventing the growth of the microorganisms. Bactosine can be used as a food additive and as an alternative to the addition of bactosine to foods. So bactosines may produce them directly in the food as well as the starcher culture or a co-cultural activity. In a present study, we determined the shelf life of milk by using the bactosine which is produced by the Lactobacillus pentosis B25 as a food preservative. Materials method for the biopreservative efficiency of bactosine we determine in raw milk and pasteurized milk. Each class contains the 50 ml raw milk and pasteurized milk. Out of that, one is control and without the Bactosine, and one is sample with Bactosine. For each type 50 ml sample used, the Bactosine was added at a 5% level and refrigerated for 2, 4, 6 and 8 and 10 days for the raw milk, and a maximum up to the 2 months, that is for the pasteurized milk. For raw milk every after 1 day, and for pasteurized milk every after 6 days. The quality and stability of milk we studied with respect to the some following points like colony count, then pH, clotting test, alcohol test, acidity test, refrigerant test, and methylene blue reductase test. According to the test, the result and discussion of this practical, the application of Bactosine as a food biopreservative in raw milk. According to this observation, the raw milk shelf life increases up to the 8 days. In a coagulation formation, the coagulation form after the 4th day, and order also obtained after the 4th day. Then clotting test in alcohol test also observed after the 4th day. This graph shows the pH and lactic acid fermentation. In a pH and lactic acid fermentation, the pH will be pH will be decreases, that is in acidic condition, obtained after the 4th day, and lactic acid concentration also increases after the 4th day. That is, this raw milk was stable up to 4 days and then decreased their accuracy. Then in a quality control studies of raw milk according to the refrigerant test and the lactic acid test as well as the methylene blue reductase test, according to this observation, the milk was stable up to 4th day, and their accuracy decreases from the 5th day and end up to the 8th day. These are the photoplates of the refrigerant test of raw milk as well as the refrigerant test of pasteurized milk. Then application of this Bactosine as a food biopreservative in a pasteurized milk. According to this observation, the pasteurized milk, the shelf life was increases up from 24 to the 42 days. Means pasteurized milk was stable up to 42 days. In a coagulation test, oil formation test, clot on boiling test, and alcohol test, there are observation occurred from 24 days and stable up to the 42 days. And pH and lactic acid concentration also observed up to the 42 days and stable up to 24 days and proceed up to the 42 days. Then next one is the quality control studies of pasteurized milk as a refrigerant test, lactic acid test, and methylene blue reductase test reduction. So according to this observation, the milk was stable from 24 to 42 days. That is, according to the observation of the pasteurized milk, by using the Bactosine, the shelf life of pasteurized milk increased up to the 42 days. These are the photoplates of methylene blue reductase test and methylene blue reductase test of the raw milk and the pasteurized milk. Now conclusion of this practical, these Bactosines have the tolerance to the wide environmental factor and it could be used as a biopreservative in a food industries. That is, the milk increase their shelf life up to 8 days for raw milk and 42 days for pasteurized milk. Thank you.
Now, we have Mrs. Sonali Joshi, who's presenting a topic to study the antimicrobial effects of citrus limata peel extracts on different types of bacteria.
Good afternoon everyone. I, Sonali Joshi, Assistant Professor, Thakur Shyam Narayan Degree College. My today's topic of presentation is to study the antimicrobial effects of citrus limata peel extracts on different types of bacteria. We know there are the different types of plants, herbal plants in India. So the World Health Organization estimates that there are the 80% of the population still depends on the herbal medicine. What is the reason behind this? That there are that herbal varieties, that herbal medicines are cost effective. So that availability of medicinal plants is not a problem, especially in the developing countries like India, which have reached in the agroclimatic, cultural and ethnic biodiversity. And main thing is that India is the large producer of medicinal herbs. So we have the lots of botanical gardens, and there are some varieties like the sweet lime, that is the tasty, juicy, fruity, and it belongs to the family Rutaceae. And the botanical name is the Citrus limata. That the study was done with the following objectives: to access and compare the in vitro antibacterial properties of different extracts of citrus species against the various pathogens. Then we take up various pathogens and we study the effect of citrus species on their pathogens, and we check the minimum inhibitory concentration of each extract of both the plants against each pathogen, with the view of searching a novel extract as a remedy for different types of the pathogen. There are the materials and the methods. We test citrus species for the cannins, for the saponins, for the flavonoids, for the terpenoids, for the alkaloids, and for the phenol. So we check the qualitative analysis of citrus species on various phytochemical constituents. Then we check the antimicrobial activity of citrus species on different organisms like the Staphylococcus aureus, Salmonella typhi, Klebsiella pneumoniae, and the Pseudomonas aeruginosa. For this, we use the agar well diffusion method. We first prepare the plate using a 4-plate method, 4 technique method for the different organisms, and then by the agar well diffusion method using the different compositions of citrus species, we check the minimum inhibitory concentration on various organisms. So the 4 different types of organisms were inoculated by the 4-plate technique, and various concentration of extracts, that is the 5 mg per ml, 10 mg per ml, 15 mg per ml, 20 mg per ml, and observe plates. Then we incubate the plate and observe the result. After incubation, zone of inhibition for each extract was observed. There is the observation and result. We do the test for the phytochemical constituents like the tannins, saponins, flavonoids, terpenoids, alkaloids, and the phenol. In the tannin, we observe the brownish green or the blue-black coloration. Means there is a presence of tannin. For the saponin, if there is a formation of emulsion and we observe the formation of emulsion, that is the saponin is present. Yellow color also forms, that is a taste for the flavonoids. For the terpenoids, there is a reddish brown color is formed. That is the terpenoids are the present. For the alkaloids, there is a sweet lime precipitate is formed. That means the alkaloids is present. Phenol, deep blue black color is formed. Then means there is a presence of phenol. We check this we check for the different organisms also, for different pathogens also. That pathogens which we use were found to be resistant against extracts of citrus limata. The concentration of all extract was significantly associated with the mean of zone of inhibition. Means as the zone of inhibition is small, then that organism may be resistant to that citrus species. Then if the zone of inhibition is large, then that organism will be sensitive to the citrus species. An increase in the zone of inhibition was observed with increase in the concentration of the extract. The minimum inhibitory zone of citrus cimetaphyll range between the 13 to 16 milligram per ml against both the dental caries pathogen. These are the some photos. We can use the peel of, we can recycle the peel of fruit. That recycling of fruit waste is one of the most important means of utilizing it in a number of innovative ways, aiding new products and meeting the new requirement of essential products required in the human, animal and the plant nutrition as well as in the pharmaceutical industry. So that citrus cimetaphyll's extract demonstrated in vitro antimicrobial activity against the pathogen. We can also prepare some products like the hand wash and the sanitizer, which is used to reduce the effect of pathogen on body. This is all about. Thank you all.
Next, we have Ms. Navya Shetty, whose topic is comparative study of antibacterial properties of neem and tulsi leaves against Pseudomonas aeruginosa and Klebsiella pneumoniae.
Warm regards to one and all present. Myself, Ms. Navya Shetty, with my presentation partner, Ms. Madalsa Patil. And today, I'm present here to give a presentation on the topic Comparative Study of Antibacterial Properties in Azadirachta indica and Osmin sanctum against Pseudomonas aeruginosa and Klebsiella pneumoniae. Before getting started with the aims, methods, and results of this research, I would like to give a quick introduction on what is antimicrobial resistance and how it is affecting health of millions of people worldwide. Antimicrobial resistance is a microbe's ability to resist the effects of medication that once successfully treated the disease. Antimicrobial resistance is related to the spread of transmissible plasmids and acquisition of resistance genes that occur by horizontal gene transfer. Certain bacteria modify their cell membrane poring channels, preventing the antimicrobials from entering into the cell. This strategy has been observed in many gram bacteria, such as Pseudomonas and Klebsiella species, against drugs such as imipenem, aminoglycosides, and quinolones. Eradication of Pseudomonas aeruginosa has become increasingly difficult due to its remarkable capacity to resist antibiotics. Strains of Pseudomonas aeruginosa are known to utilize their high levels of intrinsic and acquired resistance mechanisms to counter most antibiotics cult. Klebsiella pneumoniae is a gram-negative opportunistic bacterium that causes infections in hospitalized or otherwise immunocompromised individuals. Currently, Klebsiella pneumoniae is showing a high resistance to a broad spectrum of drugs. Now let us see what are the factors influencing antimicrobial resistance. Overuse of antibiotics, synthetic drugs over a long period of time. Evolution exhibited by the microbes and adaptation of these microbes to antibiotics. Now let us see what role Ayurveda plays in this epoch of antimicrobial resistance. Ayurveda focuses on decreasing the spread of disease, reducing antibiotic consumption by people on community level, and proper care after the antibiotic use. One of the most important factors here is strengthening the host body. Natural herbs like Azadirachta indica belonging to family Meliaceae and Tulsi belonging to the family Lamiaceae have been used for medicinal purpose since antiquity in India. Azadirachta indica has complex of various constituents including nimbin, nimbidin, nimbuloid, and luminoids, and such types of ingredients play a role in diseases management through modulation of various genetic pathways and other activities. Having detoxifying properties, the Tulsi purifies blood of any toxin that might be present. Tulsi may well provide protection from radiation poisoning. Now let us see the aim, methods used, and the results of this research. The aim was to compare the antibacterial properties of traditional herbs like Azadirachta indica and Osmin sanctum against the bacteria Pseudomonas aeruginosa and Klebsiella pneumoniae. Antimicrobial test was performed using agar-well diffusion method. After inoculation of the bacterial culture, wells were dug and the samples were loaded. After an incubation period of 24 hours, the zones of inhibition were observed and conclusions were drawn. Here we can see a plate showing Azadirachta indica zone of inhibition against Pseudomonas aeruginosa, and another plate showing Osm sanctum zone of inhibition against Pseudomonas aeruginosa. From the results obtained, an observation table was drawn and all the observations were jotted down. Here we have a graphical representation showing antibacterial growth identification. Here we have a chart showing inhibitory zone diameter. From the graph and the pie chart, it can be said that Klebsiella pneumoniae showed high resistance to neem. Results and discussion. Azadirachta indica showed a bigger zone of inhibition against Klebsiella pneumoniae than against Pseudomonas aeruginosa, whereas Osm sanctum has proven to be more effective against Pseudomonas aeruginosa than Klebsiella pneumoniae, based on the pie chart drawn. From this research, we can conclude that Azadirachta indica is more antibacterial against Pseudomonas aeruginosa and Klebsiella pneumoniae compared to Osm sanctum. What are the key conclusions drawn from this research? Consequently, the need of developing potent yet safe antimicrobial drugs against the antibiotic-resistant bacteria is arising, and due to the rich plant diversity and phytochemicals containing secondary metabolites have antimicrobial activity, it can act as a great alternative for synthetic drugs prescribed in allopathy due to its inexpensive cost and comparatively fewer side effects. We can use these plants as a special formulation for preventing and curing various infections and diseases. Plant formulations created synthetically in labs can be used in food and pharmaceutical industry effectively. With this, I would like to conclude my presentation. Now, I would like to thank our Professor Mr. Kunal Thakur for giving us this opportunity and guiding us through and through. Thank you.
Next, we have Mr. Aditya Dhobale, whose topic is isolation and identification of microorganisms from various drinking sources.
Warm greeting to one and all present here. My name is Aditya Dhobale, and my research topic is isolation and identification of microorganisms from various drinking water sources. And I'm presenting under the guidance of Dr. Bharti Wadekar ma'am. Introduction. Access to safe drinkable drinking water is one of the basic and needed amenities of humankind, especially in civic areas of the world because of the high consumption pattern of the large population inhabiting these urban and developed areas. Among the many sources of water, groundwater is considered to be the safest source of drinking water in urban as well as rural areas. According to the current reports of the World Health Organization, about 80% of all conditions in the world are directly or indirectly connected to the impurity of water. Water in its natural state is taintless, odorless, and free from disease causing microbes. The pH of water ranges from 6.5 to 8.5. The water is considered as drinkable water or potable water. Water quality in Mumbai. In the year 2019, Mumbai topped the ranking released by Bureau of Indian Standard Base for quality of tap water, with over several other major cities of India falling apart and lagging behind from Mumbai. This test was conducted in order to get a raw idea of drinking water available in majorly populated cities. This month, the BMC has bagged the Jal Nirmalata Award for 2019 to 2020 from the Ministry of Consumer Affairs for Purity of Water for their remarkable work. The lakes that give water to Mumbai are now about 99.2% full, and this is 1% further than a time ago and 1.4% further than 2 times ago. Lakes that supply or act as a source of drinking water are Vihar Lake, Tulsi Lake, Modak Sagar, Tansa Dam, Vatsa, Middle Vaitarna, and Upper Vaitarna. Indicator organisms, as it is practically impossible to test for every possible microbe present in water, and there is no single index organism that can widely be used for all purposes of water quality surveillance currently. Here are some commonly found microorganisms: coliform bacteria, Salmonella, Pseudomonas aeruginosa, Escherichia coli, Klebsiella pneumoniae, and Vibrio cholera. Methodology. Collection of samples. 5 samples were collected from 5 different locations. Sample 1 was collected from railway station. Sample 2 was collected from local snacks corner. Sample 3 was collected from bus depot. Sample 4 was collected from college canteen. And sample 5 was collected from home's water filter. Isolation and enrichment of samples. 0.1 ml of sample has been added in 10 different 10 ml broth named as nutrient broth, EMB agar, Myconki agar, mannitol salt agar, and centromite agar. We carried the dilution technique for getting isolated colonies. After 24 hours, several colonies were spotted and streaked on different media such as EMB agar, Maconchi agar, mannitol salt agar, and centramide agar. These are the streakings on nutrient plates. Identification of microorganism by gram nature and biochemical technique. Morphological and cultural identification. Colony was streaked and activity was gram stained microscopically for cellular morphology and gram stained phenotype for various parameters like shape, size, color, elevation, margin, surface, etc. Biochemical characterization. Catalase test was performed by inserting loop full of colony into tube containing 3% hydrogen peroxide. A citrate utilization test was carried out by Costa citrate agar and assay for nitrate reduction were performed. IMBIC test was also performed and ability to ferment various other carbohydrates were evaluated using sugar fermentation medium broth supplemented with filtered sterilized sugar solution to a final concentration of 1 weight by volume and 0.004% phenol red. Results. Collection of sample. The pH of sample has been shown in the table below, which range from 6.8 to 7. Isolation and enrichment of water sample. Total 108 different colonies were obtained on nutrient agar plate from 10 from different samples. Then out of 108 bacterial isolates, 35 were found on EMB agar, 14 on mannitol salt agar, 40 on Maconchi agar, and 19 on centramide agar. Identification of microbe. Following table shows the gram staining of the microbe, of which the main highlight is that organism is gram-negative with 0.5 to 0.8 mu meters in size and being green in color. Biochemical testing of bacteria. Several tests has been carried out like oxidase test, catalase test, nitroreduction test, gelatin hydrolysis test, urease test, and from Invixet, indol test, VP test, methyl red test, citrate utilization test has been carried out, which of which results has been shown as following. From above table, we can conclude that the microbe we are having is none other than Pseudomonas aeruginosa. Ruhi Rawat and A.R. Tsdc conducted similar experiments in Allahabad and found that it was well within the guidelines of the WHO, but some district samples showed contamination which could be eliminated by simple heating. Similarly, Sherry Masson conducted similar experiments on water quality of water bottle being sold in major cities like Mumbai, Delhi, and Chennai and found the presence of many harmful polymers like polypropylene and nylon. Also, Lice and Versa collected samples from Dois Corregos, Itapoi, Peroge in the Midwest of Sao Paulo state, Brazil, with having 7.6%, that is 19 samples out of 251 were contaminated. References. So I took 27% of the references from America, 5% from South Africa, 23 internationally, and 45 from India. I would like to thank our Dr. Bharti Wadekar and our college, Thakur Shamnaran Degree, for giving such a golden opportunity to showcase and present our research papers and guiding through the way. Thank you.
Now, I call upon Ms. Meenu Baljeet Jaiswal, whose topic is Estimation of Biological Oxygen Demand BOD of water samples from Daman, Nawapur and Murbe.
Myself, Meenu Baljeet Jaiswal. My topic is estimate the biological oxygen demand, that is BOD. This topic is suggested by our respected Kunal Thakur sir, and I have studied a lot about the BOD. So I put all of my effort in the BOD research. Abstract: the BOD is one of the most widely used criteria for the water quality assessment. It provides information about the readily biodegradable fraction of the organic load in the water. However, this analytical method is time consuming, and the result may vary according to the laboratory, primarily due to the fluctuation in the microbial diversity of the inoculum used. The aim of this experiment is to measure the BOD and DO of the water. Principle of the BOD is expressed as the weight of the oxygen consumed per unit volume of the water during 5 days at 20 degrees Celsius. Water sample is diluted by the dilution solution, sometimes contain bacteria. This sample is incubated during 5 days at 293 degree Kelvin and consume. The amount of oxygen is measured. Key of the BOD is BOD sewage treatment plant wetland and their ecological significance. Introduction of the BOD represent the amount of the oxygen consumed by the bacteria and other microorganisms while they decompose organic matter under aerobic condition at specific temperature. When you look at the water in a lake, the one thing you don't see is oxygen. In a way, we think that the water is opposite of the air, but the common lake or stream does not contain small amount of oxygen in the form of dissolved oxygen. I took 3 different water samples. First, Daman. As you all know, Daman is an union territory state of the India. There are various kind of big industries. Most of the industries have their own ETP. From some of the industries have waste rainwater mixed with the river and lake, etc. Here are some image from the Daman Ganga. That is restoration of river choked by the toxin. Nawapur is a village in the Boisar of Maharashtra, and it come under the MIDC. Many of the fishes die in the Nawapur due to the wastewater from the company. Murbe is a village in Boisar of Maharashtra. The Murba come under the close to the MIDC and Tarapur. Here is the beaches that are contaminated by all the industries situated near the Murbe. Material and method neutralization of sample. pH of diluted sample should be adjusted 7.0 plus or minus 0.2 before incubation. Procedure, we have to check first pH by using the calibrate pH meter, and if it is the pH is higher than the 7.0, we have to add 1 N of sulfuric acid, and if it is low, then we have to add 1 N of sodium hydroxide. pH is used by checked by us by the water sample of the water sample, and the result are neutralized. Removal of chlorine content is the chlorine is the strong oxidizing agent, and it can be inhibit the microbial growth. During wastewater BOD analysis, chlorine can be removed by adding sodium sulfide of the solution. We have to prepare some of the solutions. First, preparation of phosphate buffer solution in 1000 ml of distilled water. Second, we have to prepare alkaline iodide as an entry agent in 1000 ml distilled water. Third, we have to prepare this water dilution. The dilution water for wastewater analysis must be free from the organic content. Dilution water can be prepared. We have procedure of the BOD of water is take 300 ml of BOD bottle and add 10 ml of sample to the 2 bottle and fill the remaining volume with the dilution water. Fill remaining 2 bottle only with the dilution water for applying. Immediately close the bottle when filled, and there should be not any air bubble in the bottle. Mark the bottle as the place's name. Test of DO. Add 2 ml of 36.4% of magnesium sulfate solution, inserting the tip of the pipette tip into the sample because the drop of the solution can allow inserting the oxygen into the solution. Add 2 ml of alkali iodide reagent by above method. Allow the reacting solution with the oxygen present in the sample. When precipitate are settled down at the bottom, add 2 ml of concentrated sulfuric acid by placing the pipette tip very near to the sample surface. After adding the sulfuric acid, we can see there is a cloud formation. After disappearance of the cloud formation, we have to add starch indicator and immediately titrate it with 0.025 and sodium thiosulfate until the blue color disappear. When the blue color disappear, we have to incubate the water samples at 20 degrees Celsius for 5 days. After 5 days, we can see the automatically color has been changed. This is the observation table of day 1 and day 2. This graph indicates the BOD of the water sample. Result of the BOD of distilled water is found 0.0, which means it is extremely good because distilled water is purified, and BOD level of wastewater was found to be 3.7762 mg per liter, which means the water quality is fair because it contains a lot of pollutants. As conclusion, the water source near Daman Industry Lake has been polluted due to the incorrect handling of human activity. The recommendation to solve the problem is to apply water treatment to the water source and to prevent incorrect handling of human activity at the goal field. Then our hypothesis of this experiment is expected. This is the chart of the water quality.
Next, we have Ashwati Nair, whose topic is estimation of antibacterial properties in hair dye.
I, Ashwati Nair from Thakur Shamnarayan Degree College, is going to elaborate on a topic which is a comparative study of antibacterial properties in herbal and non-herbal hair dyes against Klebsiella pneumonia and E. coli, with my co-partner Shilpa Nair working under the guidance of Assistant Professor Mr. Kunal Thakur. The topic was chosen to enlighten about the effectiveness and composition of hair dyes. After hair graying is common after the age of 30 and most people rely on hair dyes to cover their grays. Nowadays, teenagers go through premature hair graying because of their unhealthy and stressful lifestyle. Hair graying is a real issue between all age groups as and the concern for the right product can be hectic and confusing. Weak hair follicles and decreasing melanin levels can cause hair graying, which can be controlled and at an early age. Hair nutrition in the scalp is observed by the bacteria, which in turn can result in dry scalp, dandruff, itching, and inflammation, etc. All these factors can be major issue resulting in graying of hair. Choosing the right product to treat hair is very essential, and the number of options in the market can put oneself in a loophole. The option in the market is mostly differentiated into two, herbal and non-herbal. So for herbal hair dyes, we chose Safe to Shine and Black Rose. And for non-herbal hair dyes, we have chosen Garnier and Godrej for the particular research. Herbal hair dyes are mainly made up of henna leaves, which are washed, dried, and ground. Henna is mild and gentle on hair strands as compared to any other dyes, as it doesn't penetrate into the hair cortex and gives radiant color without changing the composition of hair follicles, whereas non-herbal hair dyes are made up of hydrogen peroxide, paraphenyldiamine (PPD), and parabens. Some hair dyes also contains ammonia so as to sustain the hair color for longer period. High level of PPD can lead to severe dermatitis, asthma, as well as cancer. There is a chemical known as toluene used in hair dyes and nail pens, which can lead to birth defects, pregnancy losses, and neurotoxicity. So as to choose a hair dye which will not harm our hairs as well as do its job can be quite challenging. Thus, this experiment works on the estimation of antibacterial properties in it and also compares the effectiveness of herbal and non-herbal hair dyes. The two recommended methods for this experiments were test diffusion methods and the cork-borer method. The method that is used for this experiment is cork-borer agar well diffusion method. This method was used so that the sample was loaded in the well reacts better with the sample and shows visible zone of inhibition. 1 ml of saline solution was tipped out of both E. coli and Klebsiella pneumonia and was poured into the agar in aseptic zone. Then it was spread equally to all sides with the help of a spreader and left aside for settle for 1 hour, and later later the wells were made with the sterile cork borer in each quadrant. Later the samples were added to well with a spatula and incubated for 24 hours. After incubation, zone of inhibition was observed in both agar plates. Inhibitory zone diameter was measured in order to plot graphs, bar diagrams, and pie charts or pie diagrams to understand the statistics precisely. Thus, it was concluded that herbal hair dyes showed a visible zone of inhibition compared to non-herbal hair dyes, therefore stating the herbal hair dyes are absolutely safe for the hair and regular use of it can help in strengthening hair follicles and root firmness. Thank you.
Now, I call upon Ms. Lovelin Mandal, whose topic is estimation of antibacterial properties in paint and pen.
I am Miss Lovelin Mandal and my teammate Miss Vandana Sharma from H, working under guidance of Assistant Professor Mr. Kunal Thakur. My topic is estimation of antibacterial properties in paint and pen. I'm starting with my abstract. The wall is one place that will allow the insects and disease to stay around because the environment is not clean on the outside. Pens are something that we come across every single day. Although they are quite useful, they don't get nearly as much attention as they deserve. Most pens transmitting germs or viruses between the peoples. Thus, this experiment focuses on whether paint and pen estimate its growth against the three most common microbes found on the skin, example, E. coli, Staphylococcus aureus, and Aspergillus niger. Starting with my introduction, paint. For many thousands of years, paint has been used to decorate the spaces we live, work, and play in. This, combined with the increased prevalence of antibiotic resistance and reports the cleaning agent are having a reduced effect on microbial colonization, is why leading a global brand are looking to source effective antimicrobial paint additives that can be integrated into their paint products. Pen bacteria can thrive and survive on surface for many weeks, if not months. Your favorite pen is expression used throughout the day. It gets placed on many different surfaces. We tuck them behind our ears, use them to pin up our hair, and we may even chew them thoroughly. Inoculation of bacteria, E. coli, Staphylococcus aureus, and Aspergillus niger were isolated and incubated in saline solution and kept in incubator for 1 hour to observe a med growth. After incubation, the saline suspension was pipped out from sterile 10 ml pipette and loaded in agar plates. The bacteria was spread equally to all sides using a sterile spreader. Sample loading using quadrant method, wells were made in the agar plates with a sterile cork borer. The quadrant were numbered and the sample were loaded into the Priestley. Observing growth, the agar plates were kept in incubation for 24 hours. After incubating, it was observed that ancient oil paint showed visible zone of inhibition, whereas in Luxor antimicrobial pen has minimal in two of pathogens and yet near to negligible zone of inhibition in one of microorganisms. Method 1. Pour autoclave nutrient agar in 3 Petri plates in aseptic zone and let it cool down for 20 to 30 minutes. Make a quadrant base and after 1 hour, sterile the cork borer and make wells in each quadrant. Incubated the agar plate for 24 hours. Day 2, measure the diameter of zone of the inhibition given by both the sample. Plot the standard graph of the zone in inhibition. This is the observation table for ancient royal paint and Luxor antimicrobial pen, the bacteria which we used. This is a result for paint. This is a result for pen. And this is a graph throughout. Result after incubation, a zone of inhibition was observed around the well of each sample. Anti-royal health shield antibacterial paint showed amazing effect as compared to the pen showed visible zone. Thank you.
Next, I call upon Mr. Abhishek Jha, who's presenting a topic on scale-up in studies on production of bacteriocin from marine mutant strain of Lactobacillus pentosis B25.
So this is Abhishek Jha from Thakur Shamnaran Degree College, that is in Mumbai, Chandivali. So this is my research topic. The name of the topic is Scale-up Studies on Production of Bacteriocin from Marine Mutant Strain of Lactobacillus pentosis B25. So this research is done by me under the guidance of Dr. Bharti Wadikar in the Department of Microbiology. So the abstract for this is the bacteriocin concentration was increased and viability of the cell and mutant strain of Lactobacillus pentosis B25 was maintained till the end of the fermentation. So in this we'll be going for the continuous batch fermentation. So we'll be checking of the production of the lactic acid by the Lactobacillus pentosis strain. So, however, the industrial application of the batch production of the bacteriocin using batch fermentation has worked in commercial level. In batch fermentation, sugar consumption was equally initiated after 9 hours and led to the biosynthesis of lactic acid, which obtained up to 1.14% at the end of the fermentation. The pH was gradually decreased and the lactic acid concentration was increased. So let's see more about this research work. So the introduction that goes with the demand for foods with minimum processing of foods without chemical preservatives fast gaining moment in view of increasing concerns for health. It is well known that various pathogenic and spoilage microorganisms already present in food and could survive and multiply when there is minimal process or in absence of preservatives. So preservatives is one kind of thing that helps in the preservation of food. That could be anything. So in this respect, natural biopreservatives such as bacteriocin are non-detrimental to human health have surfaced as an alternative to process and the use of chemical preservatives in food. So instead of using chemicals, we are trying to use of bacteriocins which are made with the help of bacteria. The use of bacteriocin and the producer stain are of particular interest to the food industry since they may help to ensure the microbial safety of the food product, and it could also go with certain checking of the foods and FSSAI norms. Also, the materials and method that is used is the first is the batch fermentation, in which we have used bacterial strain, the maintenance of the bacteria, the inoculum preparation, and the media that has been used. Then the second is the fermentation experiment that we have carried out in our lab with a aluminum flask, and also the amount of the media that we have used and the strains, and also the analysis of sample, how the sample has been analyzed with different concentration that we have checked on the daily basis. Also, we have done with the optical density of the same. Also, the statistical analysis graph or the thing that we could plot. Now we have done all the things. The most main thing is the result and the analysis and discussion. Scale-up studies on bacteriocin production by batch fermentation is that the fermentation profile of mutant strain of Lactobacillus pentosis B25 with batch fermentation is 500 ml fermentation medium via MRS medium. Okay, the medium that used is MRS. During the first 6 hours of the cultivation, the mass increased up to 1.7 gram per 500 ml. And if you compare it by liters, then it is 3.4 gram per liter. If you go for the liters. That's a variable result. And then the culture passed to exponential phase. So we are going with different phases that bacteria have to go with their lifetime. So most of the bacteriocin was accumulated in the exponential phase. That is the most higher phase where the bacteria used to grow well and stable up to the end of stationary phase. So it was stable up to the stationary phase where all the material was getting to them. Sugar consumption was equally initiated up to 9 hours and led to the biosynthesis of lactic acid. So they used the sugar and fermented the lactic acid, which obtained up to 1.14% at the end of fermentation. The pH was decreased as the production of lactic acid increased and the concentration increased. The maximum biomass and bacteriocin production reached 4.65 gram per ml, and if you compare it by liters, that is 9.3 and 34440 milliliter respectively after 18 hours. Corresponding the cell viability obtained 2.128 OD at 620 nanometers when we go for the spectrophotometer observations with the counting biomass of the production and the viable count of the cells. Also, this is the diagrammatic representation, or we could say about the statistical review. There is the sugar concentration that is high in the end. Then after 9 hours, they used to start the degrading of the sugar. Then the quantity or concentration of the sugar reduced gradually as the bacteria started using and utilizing it to produce for the lactic acid. Also, the second figure is and first figure we also had the pH, biomass, and lactic acid concentration. So it was all vice versa. And also in figure 2, we have batch fermentation profile containing antibacterial activity and protein concentration and cell viability. When we go for the same, we had the zone of inhibition, also protein concentration that increased gradually, and also the cell viability. So what this conclude? From the result of batch fermentation on bacteriocin production, it is concluded that the bacteriocin concentration was increased and viability of the cell was maintained till the end of the fermentation when compared with the free cell fermentation, considering the high yield and stability of the bacteriocin even in the batch fermentation. It is essential to scale up the process as a continuous system, which can give much better yield of bacteriocin. So we will definitely try to have the different things. And also there are my few references mentioned in this. So this was my whole paper or research work that I have been done with the help of my ma'am, Dr. Badekar. And I'm also thankful to all our teachers, our coordinator, Mr. Uday sir, then also Kunal Thakur sir, Sonali Miss ma'am, Ashwarya ma'am, also our lab assistant Abni sir, and most not forget about the principal, Polari sir. So thank you.
I call upon Ms. Kushi Tripathi, whose topic is lipase catalyzed for biodiesel production.
Hi, Kushi Tripathi from Thakur Shamnaran Degree College, and my kind name is Mrs. Sonali Joshi. And my topic is lipase catalyzed process for biodiesel production. Introduction. As of late, incredible current utilization rate, the accessible inventory of petroleum products might keep going on for greater than 50 years. In expansion, expanding carbon dioxide discharge, value of copying petroleum products set tension on the biological cycle and may account for worldwide environmental change. In this specific circumstance, biodiesel as an elective fuel from sustainable sources which contain phenol-saturated fats and fatty acids can give incomplete esterification of F-prefix or trans-esterification of TGs. Protein engineering strategies, despite the fact that lipase got from normal sources can be utilized in biodiesel creation, they ordinarily miss the mark on advantageous elements that are reasonable for modern scale responses. In particular, normal lipase at greatest reactant exercise. The temperature is 30 to 50 degrees Celsius. At these temperatures, The trans-esterification response has low reaction rate, which makes the interaction tedious and less monetary cutthroat. Expanding the working temperature scope of lipase by further developing thermostability, thusly a basic part of lipase designing. First use explain crystallographic investigation to comprehend the construction of rhizomucal lipase. The construction of other significant lipase types have been distinguished in ongoing years, including Bacillus thermocatalectus, Candida antarctica, Pseudomonas capreum, and Bacillus subtilis. Thermostability, since they can be deactivated to the normal denaturation. Thermostability is an important requirement for commercial lipase. In general, increased reaction temperature enhances the solubility of alcohols in oil. It promotes faster trans-esterification reaction. Thus, enzymatic catalysts used at high temperature will increase trans-esterification yield and require shorter reaction time. Several types of lipases, particularly those originating from thermophilic organisms such as Bacillus subtilis, Thermomas lanuginus, Rhizopus aurea, and Pseudomonas species, have been reported to have heat resistance to 90 degrees Celsius. The current performance of lipase, however, still misses the mark regarding modern assumption as far as long-term thermostability. Stability in organic solvents. Low solvency of short-chain alcohols in oil prompts lipase inactivation, expansion, natural dissolver with a combination of liquor and oil works on security by improving the solvency, just as diminishing the thickness of the response combination. Nonetheless, lipase can be denatured in organic solvents, and along this line, action of the lipase can be restricted in frameworks containing natural solvents. Both coordinated advancement and objective plan approaches have been utilized to improve strength of lipase within the site of natural solvents. Lipase production. Lipase are ubiquitous in nature and are found in plants, animals, and microorganisms. Among them, microbial lipase are the most commonly used in industrial applications due to their selectivity, stability, and broad substrate specificity. In spite of improvements in lipase property in recent years, the high manufacturing cost of lipase is still the major roadblock for commercialization of lipase-catalyzed biodiesel production processes. Fungi, roots like genera, Mucor, Rhizopus, Rhizomucor, Aspergillus, and Penicillin are the significant lipase-creating sources as contrasted and microorganisms are each. The filamentous parasites are considered as a beneficial methodology. Filamentous parasites enjoy a few benefits including higher plasmid duplicate number, plasmid solidiness, and higher capacity to discharge extracellular proteins. When constructed with other heterologous cells among various organism species, Aspergillus species, also Trichoderma species, are broadly utilized for lipase creation in modern application. Conclusion: lipase-catalyzed biodiesel creation from sustainable sources enjoy a few upper hands over the regular chemical catalyzed process, including lower natural worries. Furthermore, energy utilization, the low steadiness, significant expanse of lipase, notwithstanding, have been the principal obstacles for the industrialization lipase catalyzed biodiesel creation. In this specific circumstance, protein designing and further develop lipase creation framework alongside the streamlined metabolic process are fundamental to address difficulties noted previously. Both level-headed plan and coordinated advancement strategies have been effectively used to design lipase catalysts for accredited execution. Thank you.
And now I call Ms. Sohani Ghosh, who's presenting a topic on water test analysis.
Hello everyone, my name is Sohani Ghosh. Today I'm going to present a project on the topic water test analysis with the help of my teammate Tanvi Mene under the guidance of Professor Sunali Joshi. Let us understand what water test analysis is about. What is the possibly the maximum treasured natural aid of the air? Although the floor of the earth is frequently consists of water, best a small part of its mildly usable, which makes this useful resource very restricted. This precious and recent resource consequently have to be used with prudence. As water is needed for unique purposes, the suitability of its needs to be checked before use. Also, resources of water have to be monitored frequently to determine whether they are in sound fitness or not. Poor condition of our water bodies are not best indicator of environmental degradation, it is also the threat to the ecosystem. In industries, incorrect fine of water might also result in just and severe financial losses. As a result, the first grade of water could be very crucial in each environment and financial components. For that reason, water fine analysis is essential for the use of it in any motor. In order to do water test analysis, the materials required were water samples from different sites, SCDA, that is soybean casein digest agar, SDA, sevrodextrose agar, XLD, xylose lysine deoxylate plates, McConkey's agar plate, centrimide agar plates, mannitol salt agar plates, flask, pipette, and spreader. The methods were collection of samples. Water samples were collected from 4 different locations, which are Borivli, Kandivli, Dahisar, Virar. From all water samples from all the sites were collected in sterile glass bottles, brought to the laboratory, processed within 2 to 3 hours. The preparation of spread plate for identification of microbes were 4 plates of each SCDA and SDA were prepared in a spread plate with the help of spreader. 0.1 ml of water sample was spread in each plate of both agars. Preparation of medium. SCDM was prepared in 4 plugs with 100 ml of SCDB, added 110 ml of sample in it from different sites. After that, each plugs were kept in incubation for 30 to 35 degrees Celsius for 24 hours. Then agar plates were prepared to determine the microbes present in it. Identification of microorganisms through streaking. For salmonella checking, we did RBSCB, Rappaport Bacillus Salmonella Enrichment broth. 100 ml was taken in a flask with 0.1 ml of water samples and was kept in an incubator for 24 hours. After that, XLD was prepared in an loop full of water samples from SCDM were sticked on 4 plates of XLD. All plates were kept for incubation for 24 hours. For E. coli checking, we did MACB, McConkey's agar broth. 100 ml was taken in a flask with 0.1 ml of water sample and was kept in incubator for 24 hours. After that, 4 McConkey's agar plate was prepared and loop full of water samples from SCDM was sticked on 4 plates of McConkey's agar. All plates were kept for incubation for 3 days. For Pseudomonas checking, in this, 4 centrimide agar plates were prepared and then loop full of water samples from SCDM was sticked on the 4 plates of centrimide agar. After that, it was kept in incubator for 3 days. For Staphylococcus checking, in this, 4 menthol salt agars were prepared and then a loop of water samples from SCDM was sticked on all 4 plates of menthol salt agar and that it was kept in incubator for 3 days. As you can see, we prepared plates from different sites. It is Viral. This is SDA plate and SCDA. XLD agar plate and McConkey's agar plate. Centrimide agar plate and menthol salt agar plate. The next site is Dahisar. These are the plates of Dahisar. SCDA plate and SCDA plate. XLD agar plate and McConkey's agar plate. Mannitol salt agar plate. The next site is from Borivli. This is the SDA plate and SCDA plate. XLD agar plate and McConkey's agar plate. Centrimide agar plate and menthol salt agar plate. The next site is from Kandivli. This is the SDA plate and SCDA plate. XLD agar plate, McConkey's agar plate. centromide agave and menthol salt agave. Now the result, collection of samples. The location of different from where water samples were collected are shown in this table. That is Virar, Dahisar, Borivali and Kandivali. Identification of microorganisms through streaking. Microorganisms which were identified through streaking in the agaves are shown in this table. For XLD, microorganism found was Salmonella typhi. For Macrophis agar, it was E. coli. For Centromide, it was Pseudomonas. And for Mendozolt agar, it was Staphylococcus. Now, conclusion. Assessment of water quality is essential to check the suitability of the water source for designated use. Several water quality parameters are assessed and compared with the standard values to determine the accessibility of the source of the microorganism found in it. After prolonged research, the procedure for assessment of the water have also been standardized for different purposes. In this analysis, such guidelines are discussed concisely in one place for the convenience of the researchers and analysts. Thus, this may be helpful to get an overview of the water quality assessment standards and its procedure. Lastly, I would like to thank my professor Swari Joshi ma'am to help me and my team at Gandhi Mene for making this presentation and this project a success. Thank you.
Next, we have Mr. Udayban Yadav, whose topic is photosynthesis of silver and gold nanoparticles in pomegranate, peel, and bark.
Good afternoon, everyone. This is Udayban Yadav, Coordinator, Department of Microbiology, Interdisciplinary. Today's my topic is photosynthesis of silver and gold nanoparticles in peel and bark of plant Punica granatum, that is pomegranate. Photosynthesis of nanoparticles is an emerging area in plant science research. Different plants are used for this purpose, being it is the most eco-friendly and of synthesizing nanoscale particles of different salts. The plants have their potent sources of many valuable bioactive constituents, and these constituents contribute in reduction of salt in the system. A nanometer is 1 millionth of meter. Norio Taniguchi coined the term nanotechnology in 1974. Nobel laureate Dr. Horst Stormer said that the nanoscale is more interesting than the atomic scale because the nanoscale is the first point where we can assemble something, and it's not until we start putting atoms together that we can make anything useful. In present work of Punica granatum plant was taken as an experimental system for photosynthesis of silver nanoparticles and gold nanoparticles. Bottom-up and top-down approach are considered as the two most important modes of nanoparticle synthesis. Nanocages, nanofibers, nanotubes, and nanorods are the specific types of nanostructures. Nanoparticles gives to the concept of biomedical nanotechnology, bionanotechnology, and nanomedicines. Several proteins make nanomaterials suitable for biotagging or labeling. Shape of nanoparticles varies from spherical to circular, oval, cylindrical, etc. Punica granatum plant is rich source in secondary metabolites, especially polyphenols such as alkaloids, flavonoids, tannins, and also steroids, triterpenes, etc. with lots of medicinal importance. Hence, this plant was selected. Proteins and enzymes present in plant extract are responsible for the reduction of metal ions in pomegranate. Think about the materials and methods. Here, the plant material was taken as a bark and peel of Punica granatum. The chemicals, silver nitrates, chloronic acid, deionized water. The first step of preparation is a preparation of peel and bark extract of Punica granatum. 15 gram of each peel and bark powder were mixed with 300 ml distilled water which allowed it for boiling for 20 minutes. After boiling it, filter it with the Wattman filter paper number 1. Use extract for future study. Preparation of 1 millimolar aqueous silver nitrate and chloronic acid, that is gold particles. 777.441 mg of silver iron powders and 177.223 mg of gold iron powder were accurately weighed and dissolved separately in 450 ml deionized distilled water. Next step is synthesis of silver and gold nanoparticle in pomegranate peel and bark extract. 25 ml peel and bark extract measured separately, and into this add 75 ml of silver and gold iron solution separately in a jar. Jar were agitated for few minutes and then incubate at room temperature. The method we have used for the analysis is UV spectra analysis, where bioreduction of silver and gold takes place as the time period of reaction mixtures where increases. UV spectra analysis was done at the nanometer range of 190 to 1100. The next method is TEM analysis, that is transmission electron microscopy. Before TEM analysis, samples were sonicated for 15 minutes. A drop of sample was put on carbon-coated copper grids of 3 mm diameter. Then these copper grids were kept in infrared light for drying it. Then TEM analysis is done under electron microscope. About the observation and result, the data obtained on analysis of characters of phytosynthesized nanoparticles, nanoscale particles is tabulated in this topic. Table 1, which consists of for the average particle size, shape, and distribution frequency of phytosynthesized silver nanoparticles in Punica granatum, bark, and peel extract. Here the points are to be taken care of. The NTA analysis, where the mean size of silver nitrate peel extract is 59 nanometer and the mean size of silver nitrate bark extract is 61 nanometer. Then when during the NTA analysis, the particles per frame is 26.99 particles in silver nitrate peel extract and 37.66 particles per frame silver nitrate bark extract. In case of TEM analysis, shape of the particle is oval to spherical, and in silver nitrate bark extract, it is circular in nature. Under UV spectra, the study of silver nitrate peel extract is 463 is the maximum, and in silver nitrate bark extract is 400 nanometer is the maximum. About the table number 2, where the observation table for average particle size, shape, and distribution frequency of phytosynthesized gold nanoparticle in Punica granatum bark and peel extract. Here, the gold chloride peel extract contains mean size of 54 during NTA analysis, and gold chloride bark extract contains 58 nanometer during the NTA analysis, where the particle size per frame for gold chloride is 25.51, whereas gold chloride bark extract is 10.91 respectively. Then TEM analysis for the gold chloride peel extract is the shape of the particle is spherical, whereas the gold chloride bark extract shape is circular in nature. UV spectra under nanometer, gold chloride peel extract is 548 nanometer shows the maximum, and the gold chloride bark extract shows the at a 520 nanometer. The result and discussion about the table 1 observation table for average particle size, shape, and distribution frequency and phytosynthesis in pomegranate peel and bark. The same slide we have studied under the observation table. Here's the graph of the UVC spectra recorded as a function of time for the solutions prepared using a silver nitrate peel and bark of Punica granatum, where we can see the maximum spectrum. The figure 2 of UV-VC spectra as a function of time required for the solution prepared using gold chloride peel and bark extract of Punica granatum. This image is of NTA analysis, where we can find out the particles per frame and average size of the particles. This image is of NTA analysis of 25% bark extract of silver nanoparticles. This photoplate 1ABC is of 10 image of silver nanoparticles showing spherical and oval shape nanoparticles in peel extract. This photoplate 2 is of 10 images of showing circular shape nanoparticle in bark extract. This NTA analysis of particles, where we can find out the particles per frame and the mean size of the nanoparticles. And this is the 25% analysis of bark extract of gold nanoparticle. This photoplate 3 ABC is a 10 image of silver nanoparticles showing spherical shape nanoparticles in peel extract. Here's the time to discuss about the result. The present work was conducted in the bark and peel extract of Punica granatum nanoparticles by treating with 1 millimolar concentration of silver nitrate and gold chloride. Nanoparticles in pomegranate peel and bark was done with the help of UV-VC spectrometry, nanoparticle striking analysis, and transmission electron microscope. The result obtained in the present concluded as follows. In both the experimental sample, peel and bark extract of Punica granatum, it is concluded that the biomass used yields good response for the photosynthesis of silver and gold nanoscale particles. The UV spectrometry analysis revealed the spectrum status for both these samples in accordance with nature and solved use. Nanotracking analysis of silver nanoparticle indicates high frequency of nano-sized particles in bark extract compared to peel extract of the experimental plant. The average size of silver nanoparticles was more, that is 61 nanometer for a bark extract when compared to peel extract sample. The gold nanoparticle appeared with high frequency in peel extract. However, the size of nanoparticle was more, that is 58 nanometer in the bark extract sample compared to peel extract. The TEM analysis indicates circular, spherical, and oval shape phyto nanoparticles. The gold nanoparticles were circular in shape in bark extract, whereas in peel extract, sample of the shape of nanoparticles were spherical in nature. So this is all about the experiments. Thank you.
Now, I call upon Mr. Kevat Modi, whose topic is Impact of Encapsulated Marine Strain Lactobacillus rhamnosus L43 in Alginate matrices for bacteriocin production.
Impact of encapsulated marine strains Lactobacillus rhamnosus L43 in alginate matrices for bacteriocin production. Hi, I'm Kevat Modi from Thakur Shamnarayan Degree College. Under the Professor Bhati Wadekar, from Thakur Shamnaran Degree College is here to present my research work. Introduction. Cell immobilization is defined as to fix or to fit a cell to a limit area of space or with the preserve of catalytic activity. Immobilized molecules is one whose movement in space has been restricted either completely or to a small limited region by attachment to a solid surface. Immobilization of cells is useful to stabilize the activity of bioreactors in sequential operation, plasmid stability, increasing bacteriophage resistance, and lowering the inhibition of by antibiotics or salts. In present study, the feasibility of Bactosine L43 increases using calcium alginate. Immobilized cells of Lactobacillus rhamnosus L43 was investigated and the production was compared with free cells. Materials and methods. Preparation of beads. Immobilization was carried out using the wild and mutant strains of Lactobacillus pentosis B25. Cells were immobilized using sodium alginate. Modified MRS broth was used for studying bacteriocin production with encapsulated cells to increase the stability of calcium alginate matrix. MRS broth was modified. Cells were recovered by centrifugation, washed in 0.1% buffered peptone water, and resuspended in 10 ml MRS. A solution of 4% sodium alginate was prepared in distilled water, autoclaved, and cooled for 45 degrees Celsius prior to use. The entrapment method involves mixing of 2% of inoculum in 1 volume of sodium alginate. Cell suspension of bactericin production was inoculated to alginate solution and especially transferred to sterile syringe. The mixture was forced through the sterile syringe needle by air pressure dropwise into a stirred solution of 2% of 0.5 moles fused calcium chloride. The resulting beads were hardened in calcium chloride for 1 hour, wet, sieved, and surface dried in lamina air flow hood. After preparation of beads of polymer matrices, added in 50 ml of MRS broth and incubated their respective temperature with the and lastly assayed for bactericin activity by agar well diffusion method. Determination of concentrations of cell in the media. The viability of cells was counted by dilution plating on MRS agar and after incubation for 48 hours at 30 degrees Celsius. The data was expressed as CFU per ml. Determination of cell concentration in the beads. The viable cell concentration in the beads was determined by suspending 1 ml of alginate solution in 0.1 moles phosphate solution, followed by gentle shaking for 30 minutes for destruction of the beads. The suspension was submitted to decimal cereal dilution using 0.1 ml 1% sterile peptone water. 100 milliliters for each dilution was plated in duplicate of MRS agar and incubated at 30 degrees Celsius for 48 hours. The number of viable cells were expressed as CFU per milliliter by dilution plating on MRS agar after incubation for 48 hours at 30 degrees Celsius. Comparative study of bactericin production by free and immobilized cells. The fermentations with the free and immobilized cells were performed in two different Erlenmeyer flasks containing MRS broth with the optimized pH and temperature conditions with, with without shaking for 90 hours. Determination of bactericin activity. The fermentation broth was centrifuged, passed through my millipore filter, and the resulting surfactant fluid was evaluated for bacteriocin activity by agar-wall method. Statistical analysis. Each experiment and determination was repeated in duplicate. The data were examined by one-way ANOVA using Minitab 14 at level of significance of p lesser than 0.05. Results and discussion. Stability of beads. The highest stability and the antimicrobial activity found in the sodium alginate beads. Sodium alginate has eco-friendly nature as it is non-toxic and safe for nature, cheap, used simply used, and mild conditions required for immobilization. Porosity of beads limits the nutrient supply of oxygen diffusion to the immobilized cell. Photoplate A1 and 2 show the immobilization of beads and stability. Cell concentration in the beads. The immobilized beads with the size of 2 mm in diameter were subjected to determination of cell concentration. The maximum cell concentration of 39 into 117 CFU per ml. These observations were comparable to the studies conducted earlier in Lactococcus species Lactobacillus pentococcus B25 Bifidobacterium longum ATCC 15707. Bacteriocin production by free and immobilized bacteria. The batch fermentation was carried with the free and immobilized cells of both the strains in controlled temperature and the pH conditions. The bacteriocin production profiles were similar in both of the strains in the immobilized cells. Bacteriocin production peaked at 19 mm and 25.5 mm in free immobilized cells. Photoplate B has represented the antimicrobial activity of free and immobilized cells against selected pathogen E. coli. However, when compared with the free cells, the fermentations with the immobilized cells increased the stability of yield of bacteriocin and proved long-term means of producing bacteriocin in optimized media. After reaching a peak, the bacteriocin activity decreased significantly in the culture. Such results were reported for other bacteriocins, and it could be supposed that the alginate beads had a protective role separating the bacteriocin in the medium from the proteolytic enzymes in the bead. Conclusion. The immobilization of mutant strains of Lactobacillus pentosis B25 in calcium alginate beads made possible the increase of bacteriocin production as a result of the increasing of the biomass. The calcium alginate beads was found to enhance survival cells during exposure to a number of products tested possibly as a result of factors associated with the altered cell microenvironment. Therefore, immobilized cell technology has potential increasing the effectiveness of lactic acid bacteria probiotic preparations by enhancing cell resistance during bacteriocin treatment. Acknowledgement. I, Kevot Modi, would like to express our gratitude to our professor Dr. Bharti Wadekar for helping us and guiding throughout the process, and would like to express our gratitude towards our college, Thakur Sham Narayan Degree College, for letting us stage to present our research papers. Thank you.
Now we have Mr. Kamran Abbas Mirza, whose topic is the comparative study of some conventional Indian spices against pathogenic bacteria.
Hello, everyone. we'll be discussing comparative study of some conventional Indian spices against pathogenic bacteria. India is a great producer of plenty of spices. From 80 types of spices grown in the world, India grows about 50 types of spices. Spices not only improve the taste of food, but also serve as a good source for vitamins like B, C, iron, calcium, and other antioxidants. Spices are drawn from various parts of plants like bud, bark, root, flower, and fruits. They are used in medical industries, cosmetics, pharmaceuticals, perfumeries. The role of spices is to increase the palatability, and they are also used as flavoring, coloring, and preservative agents. They are also used in industries as anti-inflammatory drugs. They are used as anti-proliferative drugs, anti-diabetic drugs. They are used against cancer, arthritis, AIDS. Herbs and spices have been used for generations by humans as food, and they are also used to treat ailments. Biomolecules found in plants play a crucial role, not only to maintain the health, but also to promote the health. They have served numerous roles through history, including coloring agents, preservatives, food additives. The active photochemical derived from the spices have provided molecular basis of this action. This present study was aimed towards the antimicrobial activity of ajwain, bay leaf, nutmeg, coriander seeds, and clove against pathogenic bacteria like Escherichia coli, Salmonella typhi, Salmonella paratyphi A, Salmonella paratyphi B. Materials and methods. Spices used were clove, coriander seeds, ajwain, bay leaf, nutmeg. Test organisms that were used were Escherichia coli, Salmonella typhi, Salmonella paratyphi A, Salmonella paratyphi B. The spices were processed for crude ethanol extract. Spices were washed individually with clean sterile water and oven dried for 1 hour at 80 degrees. Each were blended in fine powder and used for extraction. The respective plant material were processed for cold extract for 24 hours at 25 degrees Celsius. 3 grams of each spices were mixed with 30 ml ethanol. The extracts were concentrated using vacuum evaporator. The extracts obtained were tested for antimicrobial activity against pathogenic bacteria. The pathogenic bacteria were brought from pathological laboratory, and pure culture was maintained in zoology laboratory of GM Women's College, Bhiwandi. All the test cultures were maintained on the blood agar to maintain the pathogenicity. To test the antibiotic sensitivity assay, agar well diffusion method was followed for primary screening. Respective cultures of pathogenic bacteria were swept on Muller-Hilton agar plates. Wells were made using sterile cork borer, 7 mm in diameter, and loaded with crude ethanol extracts of ajwain, bay leaf, nutmeg, coriander seeds, and clove. All the plates were incubated at 37 degrees Celsius for 24 hours. Zone of inhibition were observed around the wells after 24 hours. The control used was 80% ethanol for assay. The spices used were 80 micrograms per ml. The zone of inhibition was observed. Ajwain for Escherichia coli it showed 15 mm, Salmonella typhi it showed 11 mm, Salmonella paratyphi it showed 18 mm, Salmonella paratyphi B it showed 13 mm. Whereas bay leaf, nutmeg, coriander seeds, they had their own roles. Nutmeg couldn't suppress Salmonella paratyphi, whereas coriander leaves had no effect on Salmonella typhi. Whereas clove, it was quite impressive. It showed the highest inhibition zone, where it showed 19 mm for Escherichia coli, 13 mm for Salmonella typhi, 25 mm for paratyphi A, and 20 mm for paratyphi B. Control ethanol had its own play. It showed 11 mm for Escherichia coli. It didn't show anything on Salmonella typhi. It showed 12 mm for paratyphi A and 10 mm for paratyphi B. Comparative study of the spices against pathogenic bacteria had some good results. Among the 5 spices that we used, clove showed the inhibition for all 4 pathogens. Highest inhibition effect was shown by clove on Salmonella paratyphi A with 25 mm zone of inhibition. It has also been found that clove can be used as a potent inhibitor of Escherichia coli, Salmonella paratyphi A, Salmonella paratyphi B. Nutmeg extract had high inhibitory effect on Salmonella typhi with 20 zone of inhibition. From the present study, I finally conclude that the ethanolic extract of spices may be used as potential source of natural antimicrobial compound against pathogenic bacteria. This preliminary study can be further extended in determining the active compounds of the spices so that effective medicinal preparations may be created. The employment of plant extracts and phytochemicals, each with known antimicrobial properties, may be of great significance in therapeutic uses. Spices can be used as remedies for certain ailments and may be alternative to chemical additives. Finally, I acknowledge GM Women Women's College, DIP, and Sonopan Dandekar College for their kind cooperation. Thank you.
Next, I call upon Mr. Sunil Radhakrishnan Jagyasi, whose topic is biodecolorization of orangey dye by indigenous diazobacteria.
My topic is biodegradation of orangey dye by indigenous diazobacteria. The urbanization and population explosion. We can't deny from the urbanization. It's a requirement, it's a need of the time. But whenever there's urbanization, always it results in a population explosion. And these two things, urbanization and population explosion, is always become a cause of environmental pollution. When you're talking about environmental pollution, on the top priority basis, the water pollution comes in mind. The contributors for the water pollution are the industrial. That's a requirement, need of the time. It's a requirement to be compete with the other countries. In the industries, the textile industries play a very much important role. It is a main contributor for the economic growth as well as for the water pollution in terms of color and release. Earlier, there was a use of the natural dyes, but now with the increasing population, increasing customers' choice and all, we are just slowly and slowly we are moving to the synthetic dyes. Among synthetic dyes, the azo dyes are always on the priority basis because of its demerit, because of its advantages, and there's so many merits over there. But along this one, it also brings a threat to the life because of its mutagenic and carcinogenic in nature. Whatever the physico-chemical methods are available, they are unable to answer for its removal because it is always costly process, laborious, along with a secondary sludge generation. We have to discard that. The only solution for this process is a bioremediation. Among bioremediation, microorganisms always carries a top position because of its cheaper, economic, and eco-friendly, along with a luxuriant growth, easy to grow. The diazobacteria are considered for this study. The reason for that, the role of diazobacteria in soil fertility and soil sustainability is well known. Also, it is well documented for its role in a bioremediation of the tannery effluents and herbicide degradation. So here the efforts were taken for to check the degradation ability of the diazobacteria. Coming to the materials method, the azo dye as a model azo dye was selected as an orangey dye. The sample was diaffluent sample from the local industry was considered for the microflora isolation. The sample was acclimatized with increasing dye concentration. This is in order to induce or adopt the microorganisms for the particular dye. The arrangement and isolation was carried out by using selective media, Buck's nitrogen free media, which supports only the nitrogen fixers. The degradation ability was also checked by using spectrophotometric assay using this formula. Further, the isolates were screened for the utilization of dye as the carbon source along with their tolerance and luxuriant growth at the high concentration of dye. The luxuriant growth was checked in terms of growth curve. The potent decreasing isolate was identified with reference to morphology, culture, biochemical as per Berger's manual and followed by 16S rRNA gene sequencing. Finally, the biodegradation was also analyzed for its degradation process, how the things are going on during the degradation. This was checked with the help of the spectrophotometric. Coming to the results in discussion, As I told you that orange dye was considered as a model azo dye for the current study. The diaphragm sample was collected from the local industry in Ulhasnagar, district Thane. Total 24 samples were collected in a triplicate just to cover the full area of this one. The sample was acclimatized with an increasing 20% increased concentration of dye in order to induce or adopt the organism for the particular dye. This process was carried out for the 15 days at a cycle form of the 5 days. On the enrichment and isolation, 32 isolates were selected. No growth of E. coli on this media supports the selectivity of the media for the nitrogen fixers only. Among these 32, the 17 isolates were able to decolorize the dye. Coming to the story of the screening, where the following seven isolates were able to utilize dye as a carbon source, and ultimately they show the decolorization for the dye when it was as a sole carbon source in the media. Among this, you can see over here, the isolate Da17 and Da26 were the higher decolorizer compared to others. When these were checked for the further increasing dye concentration from 100 to 500, except these two isolates, 17 and isolate 26, others were unable to tolerate the increased concentration. Whereas here, with these two isolates also, with increasing concentration, the decolorization was decreased. Up to 200, it was nearly good. So further, the luxuriant growth and tolerance was checked by using 200 milligram per liter of orange dye. As I told, luxuriant growth was checked in terms of growth curve. Here you can see that isolate Da17 showed the lag period compared to 26 isolate. So always the lag period is never accepted for the commercialization of any process or product. So it was ruled out from the further studies, and only isolate Da26 was selected for the further studies. The identification, as I told you, was checked with reference to morphology, biochemical and cultural, with reference to Burgess manual, followed by the 16S RNA sequencing, and it was identified as a Klebsiella oxytoca. The biodiesel was also assessed with reference to how the process is going on. It was just analysis by using spectrophotometer at a different time interval, 0, 24 and 48 hours. Here at a 0 hours, the lambda max can be seen at a 480 nanometer, which completely disappears at a 24 and 48 hours. One more thing, at a 24 and 48 hours, the new peaks were seen. These new peaks are the supporters for the new thing, new intermediate, new products that is formed during the process. And it was documented and also studied by so many researchers that the formation of new peaks always supports the biodegradation plays an important role during the biodegradation. Finally, coming to the conclusion, potent decolorizing isolate was identified as a Klebsiella oxytoca, was able to utilize carbon source and showed the luxuriant growth at a high dye concentration. Also, the change in visible spectra supports the role of degradation during the biodegradation by diazotropes. The further studies are in progress for the optimization of parameters along with the identification of the intermediates or the product form using mass spectroscopy. This is in order to elucidate the pathway followed for the degradation purpose and also for the toxicity studies. These are some of the key references used for the study. Thank you so much. Any suggestion, please?